Today, we're starting Chapter 6, Genetics and Inheritance, and actually in Chapter 6, this is going to be Section 6.3 through 6.6. Okay, Gregor Mendel. Gregor Mendel is known as the father of genetics. He was born in the 1820s. I don't ever really expect that you know dates. I just always think it's interesting to think about what was going on in that time frame and what technology was not available. So he was like a very forward thinker as far as what was available to him at that point in time. He studied how traits were inherited. At this time, chromosomes had not even yet been discovered. He used a plant, the garden pea, which his scientific name is Pisum sativum. So Pisum is the genus, sativum is the species. And I think we've talked about this before. You underline both of them, okay? But you capitalize the first one and the genus, and then you leave the species as a lowercase. He used this plant because it was easy to grow, it matured quickly, and it produced many offspring. This plant also had male and female reproductive parts on the same plant, which allowed for either self-pollination to occur or pollination could be transferred from the male of one plant to the female of another plant in something called cross-pollination. If the pollen was compatible, fertilization may occur. So I'm going to draw you a picture right quick, okay? So, and this is only the reproductive part of a plant. So if you have, okay. Okay, so we have male and female reproductive parts on a plant. So this part here is the male, and it's made up of two portions. This portion is called the filament, and then this portion is called the anther. The anther is going to produce pollen grains, and pollen grains carry some sperm cells in them. This center portion is called the pistil, and this is the female. And the pistil has three parts. The top part is the stigma. The center portion is called the style, like women style their hair. And then this portion down here is the ovary. So the ovary contains ovules that if they get fertilized, then they become a seed. Okay, self-pollination simply means that the pollen from a plant falls on the stigma of the same plant, okay? And so he could self-pollinate it, or he could cross-pollinate, which means he would take pollen from one plant and transfer it over to the stigma of a different plant. So Mendel's experiments. He developed what is called the purebred generation or true bred, which are strains for a certain trait by growing many generations, and he called that his parental generation or his P generation. So we're going to be using this word P generation when we do crosses. Okay, so he knew exactly what was in that what that trait coded for. So for example, he would breed breed plants over and over and over, and they would always produce round seeds. Mendel could control pollination by removing the stamen, which produced pollen. So pollen carries the male gamete. He would then pollinate the female pistil with pollen from the stamen of a different plant. So this would be cross-fertilization or cross-pollination, excuse me. He worked with 7 traits. He worked pea shape, pea color, pod shape, plant height, flower color, and flower position. These traits would show either or characteristics. There would be no intermediate. So I've got a little thing to show you. So here are the traits. So the position of the flower along the stem was either axial or terminal. Axial was going to be your dominant. The height of the plant would either be tall or short. Tall would be dominant. Pod appearance, either the pod is inflated or it's constricted. Inflated would be dominant. Pod color would either be green or yellow. Green was dominant. Seed texture or seed shape is what I call it. The seeds would either be round or wrinkled, with round being dominant. Seed color, now this is kind of strange. The pod color green is dominant, but the seed color yellow is dominant. So the seeds would either be yellow or green, with yellow being dominant. And then seed coat color, the coat around the seed would either be colored or it'd be white, and so color would be dominant. You don't have to memorize these, okay? Mendel pollinated one parent to another to produce hybrid seeds. So a hybrid is defined as the offspring of crosses between organisms differing in one or more characteristics. He cross-pollinated the pea generation. He collected the seeds. He would plant them, and then he would produce the F1 generation. So when you cross your parent generation, then the next generation is called the F1 generation, or the first filial generation, and he would record his findings. Then he would allow self-pollination to occur between the two F1s. And when the new generation that comes out of that is called your F2, or your second filial. So in the parental generation, you're going to have cross-pollination, and then when you get your F1, and then when you breed back your F1 to another F1, then that is self-pollination. He worked with thousands of plants. He kept very detailed records, but his work was not accepted until the discovery of genes on chromosomes in the 1900s. And by the way, if I'm not mistaken, I think Gregor Mendel worked in a monastery. Okay, his conclusions. So some people call these laws, some people call them principles. So one conclusion he came up with was called the principle of dominance and recessiveness. So one gene may cover up the other gene for the same trait. So the gene that would cover up or mask the other one would be called the dominant trait, and the other one would be recessive. So like tall would be dominant over recessive. Then he came up with something called the law of segregation, which is called his first law. And this law says that members of each pair of alleles will separate when gametes are formed during meiosis. A gamete will receive one allele or the other. So if you'll recall, we talked about homologous chromosomes in the last chapter. So if this is a gene, this is a chromosome from dad, and then this is a chromosome from mom. And if there's a gene here for height, then on mom's there's a gene for height. And so it's saying he would either get the one from dad or get the one from mom. So a gamete will receive one allele or the other. So this law explains something called the 3:1 phenotype ratio of the F2 generation of a monohybrid cross. So we're going to do a monohybrid cross in a minute. So the law of segregation came out of studies with the monohybrid cross. Then the next thing he came up with was something called the law of independent assortment, or his second law. And it simply states that allele pairs separate independently from each other during meiosis. Different traits appear to be inherited separately. So another way of saying that is when the new organism is formed, it's not necessarily going to have all the dominant traits. It could have some of the dominant and some of the recessive to show up. So he discovered when he crossed plants for two different traits, for example, seed shape and height, that not all dominant characteristics appear together. So in other words, round seeds, which are dominant, could also appear on short plants, which are recessive. You could have round seeds on tall plants, or you could have round seeds on short plants. This explains the phenotype ratio of a dihybrid cross, and it comes out to be a 9:3:3:1 phenotype ratio. Okay, so your 3:1 is with your monohybrid phenotype, and 9:3:3:1 is with your dihybrid. Okay, genetic crosses. So terminology. So we're going to be using these terms quite a bit in our crosses. So genotype is the genetic makeup of an organism. It's represented by letters. So if you said, for instance, big T, you're assigning letters for height. So this would be the genotype. Phenotype would be the physical appearance of the organism. You use word description. So if you said big T stood for height, then the phenotype would be the individual would be tall. Alleles are alternate forms of a gene. So like an allele could be for height, so the height could either be tall or short. Those would be alleles for each other. Locus, or loci is plural, is the location of a gene or allele on homologous chromosomes. And so they've mapped them out, and they know exactly the loci of all the different genes in the human genome. Homozygous are when both alleles of a pair are the same. So if it's homozygous dominant, for instance, it will be two uppercase letters of the same type. So big T, big P, big L, big L. Homozygous recessive on the other hand, will be two lowercase letters of the same type. So like little t, or little l, or little p, p. If the genotype shows one of each, an uppercase and a lowercase, we say that it is a heterozygote or it's heterozygous. So when two alleles of a pair are not the same. So for instance, big T, big P, big L, little l. Okay, so let's talk about our first cross. So we're going to talk about a monohybrid cross, and how monohybrid crosses work. So with mono, think of mono as meaning one, okay? So we're going to just talk about one trait. So it's a cross between two individuals involving only one trait. So for instance, pod color. Pod color has two alleles. The green allele and the yellow allele. Green is dominant since we gave it the uppercase, and yellow is recessive since we gave it the lowercase g, okay? I would always give you the alleles to use, okay? Okay, so we're going to use a Punnett square to predict the probabilities of offspring. I don't think I have enough room here, so I'm going to do it on another sheet. Okay, so let's say, for instance, we know that we're working with green, which is your uppercase, and yellow, which is your lowercase. And this is going to be for pod color. Okay, so remember, your parental generation has been purebred, so we know exactly what's in it. So parental generation, P generation. By convention, you usually put the male gamete first and then the female gamete, or female individual second, not gamete, individual. Okay, so one parent is going to be big G, and we're going to cross it with the other parent that is little g. So this parent would say is homozygous dominant. This parent would say is homozygous recessive. Through meiosis, this parent will only give one of the big G's. So remember, that was your law of I lost my train of thought. Dennis, excuse me. Law of segregation, okay. And through meiosis, this parent will only give a big G, and so you show your gametes with a circle around them. And then this other parent through meiosis will only give a little g, okay? So then he would allow cross-fertilization to occur, cross-pollination, and the generation that came out of this was called the F1 generation, or first filial. And so when fertilization occurred, this big G and this little g got into the zygote, so the zygote turns out to be big G, little g. Now, what he did is he allows self-pollination to occur in the F1 generation, so it comes back and it breeds back to the same individual. And so we want to figure out what can we get out of the first filial generation to get our F2. So for our F2, we're going to do the Punnett square. Now, I may do mine a little bit different than you have learned, and that's okay. So the way you do a Punnett square is you figure out how many gametes can come out of your parent. So this parent can give a big G, or it can give a little g. And the same for this parent. So we need a Punnett square that can handle 2 across the top and 2 down the side. So I'm just going to do a regular tic-tac-toe. By convention, you put the father's gamete across the top and the mother's across the side. So the father can either give a big G in his gamete or a little g, and the mother can give a big G or a little g. If this sperm and this egg meet, then this individual would be big G, big G. If this sperm and this egg meet, this person will be big G, little g. Typically, you always list the dominant allele first. And then if this sperm and this egg met, big G, little g. And then if this sperm and this egg met, it'd be little g, little g. So from this Punnett square, we can get what we call our genotype ratio. And our phenotype ratio. Okay, so genotype, recall, genotype are represented by letters. So you would say out of the F2 generation, you could get 1 big G, to 2 big G, little g's, to 1 little g, 1 little g. So that is always a 1-to-1 genotype ratio if both of your parents started out, one is homozygous dominant and one is homozygous recessive. Phenotype ratio, remember big G is dominant, so this individual would be green. Since green masks the yellow, then this would be green and this would be green. So you would have 3 green to 1 yellow. So it's a 3-1. Okay, so once again, if one parent is homozygous dominant and it's a monohybrid and the other is homozygous recessive, these would be your ratios every time for your F2. So it's a 1-2-1 for genotype, a 3-1 for phenotype. Now, a test cross is used, it's breeding an organism of an unknown genotype, so the parent could either be homozygous dominant or heterozygous with a recessive homozygote, and that will go back and tell you what the parent is. So for instance, if you have a cross between a green pod with a yellow pod, what is the probable genotype of the green parent? Okay, so the parent could either be this or it could be this, because both of these are going to show green. So we're going to figure out which one it is, and we're going to breed it back to a homozygous recessive individual. Okay, so if it's this parent can only give a big G, right? Okay, so if you've got this parent big G, we can go ahead and put another big G, but it's redundant, and you're going to put it with a homozygous recessive individual, little g, little g. So that means that all of their offspring would turn out to show the dominant trait, so they'd all be green. Instead, if this turned out to be your parent, so that parent could either give a big G or a little g, so you do another punnet, so that parent can give a big G or a little g. This parent can only give a little g, so here's what I'm going to do on this one. I'm only going to put the little g once because it's going to be redundant to put it back down here. You can. Okay, so this would show that half the individuals in the F2 generation would actually turn out to show the recessive trait. And so a test cross is used to determine what the genotype is of the one of the parents. So you cross a homozygous recessive individual with the parent, and that's going to tell you if your parent is homozygous dominant or is a heterozygote. Okay, dihybrid cross are next. Okay, so this is a cross that's between two individuals involving two traits. So I will always tell you what the two traits are so that you don't have to come up with them. So for instance, we're going to do pod color, which we did a second ago. Green was dominant and yellow was recessive, and we're going to do it with seed shape. Round is going to be the bigger uppercase R. It's the dominant, and then wrinkled is the lowercase r. It is recessive. Okay, so we're going to do another punnet. Okay, so our P generation, so one parent is going to be homozygous dominant. So it is going to be big G, big R. And you're going to cross it with an individual that's homozygous recessive. So that is big G, little g, little r. Okay. So by convention, this is usually the male, this is the female. Now, through meiosis, the one of each of these will get in. So one of the alleles for one of the alleles for pod color and one of the alleles for seed shape will get in. So this gamete will have a big G and a big R in it. And through meiosis, this individual will have a little g, little r. Okay. So once again, you're going to have cross-fertilization occur or cross-pollination. Out of that, you get your F1 generation. And so this sperm meets this egg. So in the new individual, it's going to be big G, big R, little r. Keep your G's together, keep your R's together. Once again, by convention, you always show the dominant allele first, okay? And so then we're going to allow self-pollination or self-fertilization to occur, and we're going to breed it back to the same type of individual, okay? So now, if we want to figure out our F2 phenotype ratio, then we're going to use a Punnett square. Now, once again, look at your individual and determine how many gametes can come out of it. That's going to tell you how many spaces you need on your Punnett. So this individual can give a big G, big R. It can give a big G, little r. it can give a little g big R, or it can give a little g little r. This individual is the same, so it's going to be the same thing. So it's going to be a big G big R, a big G little r, a little g big R, and a little g little r. Okay, so as you can see, we're going to need a Punnett square that accommodates 4 individuals across the top and 4 gametes across the side. Okay, so it's going to be a big Punnett square. Make your square big enough that you can write phenotypes in it. Mine's not very tidy. Okay, so we're going to put dad's gametes across the top, mom's across the side. So he can give a big G big R, he can give a big G little r, he can give a little g big R, little g little r. Mom's same down the side. Okay, now you're going to complete the Punnett square. So I'm going to complete mine. If you need to put it on pause for a second, you can do so. So this individual is going to be big G, big R, big R. This individual is going to be big G, big R, little r. This one's going to be big G, little g, big R, big R. This one, if this egg met this sperm, that would be big G, big R, little r. This sperm met this egg, that goes here, big G, big R, little r. Sperm and egg, that would go there, big G, little r, little r. If this sperm and this egg met, that would go there, big G, big R, little r. These two meet, that's going to be big G, little r, little r. Okay, big G, little g. Okay, so once you get the genotypes in, what I would do if I were you, I'd go in and write what they each are. Okay, so this one's going to turn out to be green round, green round, green round, green round, green round. Green wrinkled, green round, green wrinkled, green round, green round, yellow round, because lowercase little g, yellow round, green round, green wrinkled, yellow round, and yellow wrinkled. Okay, so we're going to go through and count how many green round we have first. Okay, so we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. So you should have 9 green round. And then let's do green wrinkled. 1, 2, 3, and now yellow round. So that is 1, 2, 3, and then yellow wrinkled, which is completely recessive. is 1. So if you start out with a parent that is homozygous dominant for a dihybrid and the other parent that is homozygous recessive, then you would expect your phenotype ratio every time of your dihybrid F2 to turn out to be a 9-3-3-1. 9 would show the homozygous dominant trait, 1 would show the homozygous recessive, and then you would have 3 and 3 of your heterozygotes. Okay? So this is where you will stop and you will do your study guide 1. Thank you.